In this video, we will discuss about different types of glycan that are present or that occurs in the mammalian cells. So, glycans can be exist uh, on its own, such as hyaluronic acid, and glycans on other ways can exist on the proteins. These are shown as the N-glycans. These are which are usually exist or which are usually found on the asparagine side of the proteins, or we can say on at the asparagine residue. However, they are not found in any asparagine residue. They need to follow a specific sequence, that is NXT, that is asparagine, then any amino acid. After that, it should be either serine or threonine, and in this any amino acid, that is X, X should not be equal to proline. So it is found at sequence, that is NXT, where X is not equal to proline. And whereas these O-glycans are found at serine and threonine sites of the serine and threonine sites or residues of the glycoproteins on the proteins. And this is the hyaluronic acid that exists on its own without any attachment to the glycoproteins or any proteins. And this is also known as glycosaminoglycan. And these are the GPI anchored glycoproteins. In this case, we can say that the glycans are attached. Attached to the this cell surface membrane or this lipid membrane with the help of this GPI anchored that can be seen here, and these are attached to this phosphate bond. And another kind of glycans is the proteoglycans that are shown on left hand side, and it contains this chondrionic sulfate and heparanine sulfate related glycans are attached to these proteoglycans. And or we can say this proteoglycans means when the number of glycans that are attached to them is low, whereas in the case of glycosaminoglycan, number of glycans attached are very high. And these glycosphingolipids, and these are the glycans that are directly attached to the directly exist on the cell surface, and they are attached to the extracellular membrane directly, not like this GPI anchored protein where they are attached via this GPI, whereas in the case of glycophospholipids, they are directly attached to the cell surface membrane. So now let us briefly understand what are the cancer treatments are available and what are their disadvantages and what is the next possible solutions. So the conventional therapy for the cancer treatment are the chemotherapy, radiation therapy, and surgery. And the problem for these therapies are the side effects and lack of specificity and invasive procedures. That is procedures that will not cure this cancer completely, whereas some parts of tumors are left within the patients that will regrow themselves and infect the individuals again. Next therapy is about the immunotherapy, where the antibodies are generated against the specific antigen, such as this CAR T cell therapy and cetuximab, trastuzumab, and PDL1. All these immunotherapies can target the antigen that are present on the tumor cells, that are predominantly present on the tumor cells. However, the disadvantage of these immunotherapies are like they are very laborious. They require the process of production of these immunotherapies are very laborious, and due to the high heterogeneity, one antibodies cannot be applied to large populations. And another one is this therapy resistance. So with time, the host body will develop a resistance against this kind of antibodies, and that will reduce the half life of these antibodies and finally reduce the efficiency uh, towards the treatment of cancer. The next one is the small molecule inhibitor such as kinase inhibitor, for example, gefitinib, uh, sorafenib, etc. The problem for this is also similar to the other cases, that is side effects and toxicity of the drugs, and also the drug resistance, that is immune system of the patients will develop a immune response against this kind of small molecule inhibitor. Therefore, that will reduce the half life of the small molecule inhibitor, as in the case of immunotherapy, and reduce its final efficiency for the cancer treatment. So, what are the uh, next possible solutions that researchers are focusing on is about targeting glycosylation, and as in the it has been reported many times in the literature that cancer has aberrant glycosylation. uh, Cancer tumors has a aberrant glycosylation that is going on within the cancerous cells. So that will leads to the aberrant glycan epitopes that can be expressed on the cell surface membrane. So antibodies can be developed against those glycoforms, and or the some inhibitors can be used. Some specific inhibitors can be used to target those glycoantigens and trigger the immune response against the tumor cells that contains those kind of glycoantigens. And these glycoantigens are more specific in the tumor cells and compared to the healthy cells. So these are some of the cancer treatments that are presently been going on, and where the research is about to move forward to find out a next possible solution. I hope you have understood something about this cancer treatment and different types of glycans. If you need to understand more about these topics, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.